is Niklas. I'm CEO at our Census, and for the next 45 minutes, I'll also be your host and guide you through this webinar. I'm super excited today. I've been waiting for this for a couple of weeks now. We're going to touch upon the topic, finding the critical genetic needle in a 6 billion genome haystack. And I'm also glad to see that I'm not the only one who's excited, but also the number of participants and all the positive feedback we received in the last few weeks. I know that quite a few of you have been looking forward to this topic. Before we get started, I'd like to welcome all of you formally. It's a pleasure to see that we almost have 200 participants joining in from all over the world today. It's an honor to have you on board, and we will make sure to use your time wisely and to try to share as much knowledge as possible with you before we get started. Cool, so let's get straight into it. Before I present today's speaker, I'd like to give you a brief introduction on how to use the Q&A section again. So during the presentation and also after presentation, we will have time for you to hand in your questions. We will have a slight discussion round afterwards. We will try to answer as many of these questions as possible. If we don't manage to answer all of them, we will send the answers via email to all of you afterwards. So if you want to hand in any questions, you see at the lower part of your screen, in the Zoom screen, you see this Q&A part. If you click on that, it will open up the Welcome to Q&A screen. Here you can type in your question, write whatever you want, and click on Send. Once this is clicked on Send, our team will screen through the questions, we'll sort them, and then after the presentation, we will have a structured discussion about it. So that's it. If you have any questions in between, you can also use the chat to ask us if you don't know how to handle it. Our admins will see it and get back to you. Perfect. So let's get straight to it. And today's speaker, I already announced it last webinar that I'm super excited to welcome Dr. Najim Amitsiane for today. He is the Senior Director of Bioinformatics Research at our Census. He has a PhD in human genetics and has over 15 years experience in molecular diagnostics of hereditary diseases and cancer. He has also pioneered the application of next generation sequencing in the clinical genetics department of the VU Medical Center in Amsterdam. And he has discovered quite a few new gene diseases associations by utilizing cutting edge technologies in the laboratory as well as bioinformatically. And what's most exciting for me is I know his passion about deciphering the human genome. He's a geneticist through and through, and it's a great pleasure to have you on board, Dr. Najim. I'm happy to hand over to you, and you can share your presentation with us.
Thank you, Niklas, for the nice and kind words and the introduction. So what I would like to do today is I would like to go and take you through a journey from going from a DNA sample that is sent to us at our census and isolating the DNA all the way to the identifying the genetic variant that is causing the symptoms or the disease of a specific patient. So, okay. During this presentation, I will briefly discuss a little bit about the DNA, which, as I mentioned earlier, is the code of life, and then typically how whole genome sequencing analysis workflow happens in the clinical genetics setting, and therefore going from DNA all the way to the report. And then I would finish off with some examples, and then the floor will be open for some questions to which I would like to provide some answers where possible. So just briefly, so that everybody is on the same page, as the majority of you know, our DNA is stored in the cell in a way that is preserved and highly in a high fidelity manner, and going from the DNA all the way down to the proteins that eventually perform the functions that are necessary for life. There are different types of proteins that are known. There are some of which that have functional functionality, like scissors or gluing two different components to each other. There are other components, proteins that are involved in signaling, and obviously the other part which are part of the building blocks. And just that everybody is also on the same page, in our genome, so in one cell harbors what is called the diploid genome, which is part of the genome that we have received from our father, and the other ones from our mothers. And in total, it's about 6.4 billion bases, and hence the title. Of those 6.5 billion, more or less, is consisting of the coding exons. So those are the regions that are eventually actually coding for the information that is harbored in the proteins. And even though we have about 6.5 billion bases, almost 4 million are benign variants, meaning that they are present in multiple individuals and they do not have any harmful effect. And many of those benign variants are known to be structural variants. So these are variants that are not single subtle substitutions, which I will explain a little bit later as well. So the genetic diagnostics actually stands for the identification of one or a few mutant bases in a genome. It can be a single base or a huge, large chunk of the genome, of the DNA. So just to give you an example, if we take two individuals, if we compare their genomes, they would be for 99.8% identical, meaning that although they are almost identical, about 0.2% of this whole genome within those two individuals would have about 6 million of nucleotides that's different between those two. So we have 6 million different variants. As I mentioned earlier, those different variants are not harmful. Most of them are non-pathogenic, so they are either single nucleotide differences or they are consisting of large aberrations like copy number alterations. And just to bring this in perspective, if we compare the genome of the human to the genome of the chimpanzee, there's only a 2% difference between the two species. So it's actually a tiny number with regards to the differences. So when it comes to genetic testing, there are two main things that one needs to consider. So there is, first of all, a different mode of inheritance. And of those, I would like to mention a few. So we have the autosomal mode of inheritance, meaning that there is a single variant present in either the father or the mother that can be inherited from parent to offspring. So you don't need both parents to be carrier. So only a single variant can cause a disease, which can be followed in this pedigree. And then you have what is called the autosomal recessive mode of inheritance, meaning that both parents need to be carriers to be passing the disease-causing variants to the offspring. And as I mentioned earlier, you have two copies, and each of those copies that is harboring a difference, a mutation that is present in the offspring, that's when you have the disease. And then you have the X-linked dominant and the X-linked recessive, which mainly affect the sex chromosomes. And then lastly, you have the mitochondrial genome that can be affected, which can only be inherited from the mother, since only the oocyte of the mother can be passed on to the offspring. And then you have the second part when it comes to genetic testing, which is the different types of variants that can be associated with a disease. And these are mainly in the majority of the disease-causing variants caused by what is called subtle variants. And these are either substitutions, meaning a single nucleotide change from one nucleotide to the other, or deletions or insertions that are smaller than 50 nucleotide in size. And the second part are structural variants, which are in general gross deletions, meaning deletions or duplications that are larger in size than 50 nucleotides. And then you have inversions where a piece of the DNA is put back into the genome in the inverted direction. And lastly, you have translocations, which meaning that you have a chromosome where a break took place, and then to be able for the cell to survive, this break is repaired by attaching one chromosome to the other. And for these two different subtypes of variants, there are different tools that need to be used to identify those. So just to summarize briefly, so we have the single nucleotide variants as depicted all the way at the left. So this is a depiction of a cartoon of a chromosome at the top. And then you have below this chromosome at the top, the reads, the way they are aligned to the reference genome. So whenever you have a difference between the reads, the raw data that is obtained, well, that is different than the reference, then it will be called as a different. And this is how a single nucleotide variant looks like. And then you can have an indel, in this particular case, a deletion of either a single nucleotide or up to 50 different nucleotides. And then you can have a gross deletion, which is in a homozygous state, affecting a large chunk of the DNA, or in a heterozygous state. And then you can have a duplication, which is a gross duplication resulting in a gain of material in the form of DNA. And then you can also have what is called a translocation, where part of the reads are mapped to one chromosome and the other part of the reads are mapped to the other chromosome. And this is how this type of alterations can be picked up. So to give you an example that, for instance, a single nucleotide difference can actually explain a very severe disease, as is the example in this case. So we have this individual with this small boy with many severe symptoms that are depicted all over here. And all these symptoms can be all attributed to actually a single change on chromosome 18 at one position at this location that actually caused a change from a G to a nucleotide. And this affects a gene that is NPC1. And this is just some information that tells you about what type of change this particular single nucleotide substitution is causing at the cDNA level as well as the protein level. So just briefly again on NGS. Next, which stands for next generation sequencing. What is actually done there, we read the DNA almost literally. So to do this, we want to identify the relevant information from the DNA, and for whole genome sequencing that we apply here at this company is the most comprehensive genetic test that can be used at this day and age. Briefly, how the procedure looks like. We start off once the DNA, usually it is sequenced on a high throughput sequencing machine that initially actually generates just pictures. And these pictures is then transformed into what is called the raw reads. And then these raw reads need to be aligned to a reference genome using high throughput computing machines. And after this alignment, a variant detection is done, which generates certain files. And then these certain files that have information on variants need to be annotated to provide some context to those variants. And then the filtering and the prioritization of these large files is taken place by the geneticist to identify the candidate genes that could explain the symptoms. And I will try to go through those steps in the next slides. So it starts off with the DNA. So usually the DNA is a very, there are many large molecules that is obtained from samples from individuals. This has to be shared into small pieces, ironically, and then it has to be prepared to add adapters at each end, and these adapters need to be done so it can be added to what is called a flow cell that is depicted here. And after these molecules with these adapters are added to the flow cell, it looks something like this. These molecules need to be amplified to make them larger to be visualized with a camera. And once this is visualized, then cyclically, nucleotides are added to those sequences, so it can be read, and this is done in a massive parallel manner. And this is how it is done. So we have a high definition, high resolution digital camera that actually scans that flow cell that I showed you before. And in each cycle, a nucleotide is added, a picture is taken until the entire sequence is built up. And this is more or less how it looks like. We have the flow cell with the different alleles, and then we have each cycle the nucleotide is added. The camera scans through the whole flow cell to take pictures, and then all the way at the end, the final results would look something like this. So there is a stack of pictures that are put on top of each other, representing the different cycles. And once this procedure is done, we end up with massive amount of data that we need to find and identify as potentially a single variant that is potentially explaining the symptoms of the disease. So this is how the raw reads are coming from when the sequencing run is finished. So it's just a huge text file that contains some information, and this information just tells you something about the machine on which it was sequenced, so you can track the data all the way back to the machine on which it was sequenced. You have some sequence identifier, you have a barcode so that you can discriminate between the different samples. And then this is the actual information which is in the middle. That is the actual read sequence from the DNA of an individual that has been sequenced. And then finally, you have some qualities that are related to the different sequences in the middle. And once the sequence is obtained, then the sequences, what is also called reads, are aligned to the reference genome. And this is more or less how it would look like. However, since this is a little bit harsh on the eye to read, it is mapped in a different way. It is shown in a different way to only illustrate to us what is actually different from the sample of interest and the reference. So by doing so, we only display the differences between what is the reference and the sample of interest. And then once this alignment has taken place, and then we try to identify all the differences that has been identified between a specific sample and the reference genome. And this is then put in a file that is called the VCF file that obtains information about the chromosome and the position of where a difference has been identified, and then some additional information about how confident we are about the specific difference that is present in a sample. And once it's there, then we need to add some context to this VCF file, because just knowing the position and the chromosome of the variant does not tell us whether this particular variant is actually affecting a gene. And this is what is done at this step. And at this step, we provide information from different databases to this different variants. And these databases that we use, they are either related to the gene, which are depicted in the boxes in yellow, or they are related to the variant, which are depicted in the boxes in blue. So a single variant can be annotated by the information from the sequencing, which tells us something about the quality of the variant, how reliable it is that this variant is indeed present. And then we add information from databases like SNP database and GNOME, which tell us how frequent has this variant been identified in healthy individuals. And then we add information about whether this variant can affect splicing. So it can be harmful, yes or no. And then we get information as to whether this gene has been described earlier in publicly available databases, like is it published already in association with a disease, and also information from mouse experiments and other different databases that does some kind of prediction of whether a specific variant can or cannot be harmful. And these are just a handful of the different databases that are used. I don't want to go into this too much in detail, but just to illustrate that we using different types of databases to obtain as much information on specific variants and genes that we have identified as being different than the reference. And one very important aspect of this data and the annotation is obtaining information which is not necessarily related to the variant, but more specifically related to the individual, which is the human phenotype ontology. And this is information that is usually provided from the physicians in the form of the clinical description of the symptoms. And this clinical description of the symptoms is then uniformized in the form of standardized text that we can use eventually to connect the gene that is affected to the symptoms that had been described by the physician. And just to give you an example, for instance, some HPO terms, so some of those human phenotype ontology are associated with multiple genes. So meaning that one gene can be actually, if defective, can be associated with different types of diseases and therefore different types of symptoms. And at the same time, some of the genes can also have multiple HPO associations. And this is an example, for instance, of a gene. This particular gene is known when defective. It can be associated with symptoms that involve ectopia lentis, skin ulcers, and all these symptoms that are mentioned here. At the same time, if we take the emphysema symptom, this emphysema symptoms, as a matter of fact, is involved in more than 20 different genes. So if those genes are defective, the patients may have the emphysema as a symptom. Just to give you an indication, there are currently about almost 9,000 unique HPO terms, and these HPO terms are known to be associating with over 4,500 unique genes. And also to give you an indication, the gene with the most HPO terms, it's a gene that has about 750 different HPO terms associated with it. But at the same time, you also have a gene that has only a single HPO term associated with it. However, on average, each gene has about 54 HPO terms associations and with a median of 36 HPO terms. And now I would like to give you an example how the filtering procedure takes place. So after, as I showed you earlier, identifying the variants, you end up, when analyzing whole genome sequence data from a single case, you end up with over 4.5 million variants. So 4.5 million different nucleotide changes that has been identified in an individual when compared to the reference genome. So the obvious step to start doing the filtering after these variants had been annotated, and this is, I hope you understand why this annotation is so important, because you need to have some indication on, you need to reduce this number of 4,500,000 to a manageable size of candidates that could explain the symptoms. And the first analysis or the first filtering step to perform is to filter, obviously, for variants that are expected to be true. So a quality filter takes place. And then the next step is to focus on variants that are expected to have an effect on a protein. And then once this step is taken place, we reduce the number of 4.5 million to a little bit over 25,000 coding variants, because in general, those are the variants that are expected to be disease causing. And then we filter for those that are expected to be very rare in the databases of healthy individuals, because if they are frequent in healthy individuals, they are not expected to be disease causing. And once we have the rare variants, then we start filtering based on the mode of inheritance. In this particular example of a recessive disorder, we try to identify variants, at least 2 different variants in the same gene, or 2 similar variants in the same gene. And once this step is taken place, so to do this very well, you need to have 2 variants in the same gene. So it could be either 2 single nucleotide variants, or a single nucleotide variant together with a structural variant, which could be a copy number alteration. So this suggests that one variant is inherited from one parent and the other one is inherited from the other parent. And if you have siblings also sequenced, then you can use the unaffected siblings to exclude this, the presence of these 2 variants in the same gene in the unaffected individual. And then you end up with a list of candidate genes, and then we use the HPO terms that I just explained in the previous slide to only focus on the genes that are expected to be associated with the symptoms. And then we end up with the top candidates. And for these top candidates, we need to either confirm functionally if the gene is not yet known, or if the variant is not clearly expected to be disease causing. And this is where we end up with a relevant variant. However, at the stage of the top candidates, if no relevant variant is identified, we have to take a couple of steps back and then maybe come up with a different mode of inheritance, which should always be kept in mind. And then repeat the filtering approach. So now I would like to give you an example of whole genome sequencing that was performed on a single case in a recessive disorder. So in this particular case, with the symptoms that are mentioned here, which are related to neurodegeneration disorder, involving also leukodystrophy and mental deterioration, we started off with about 4.8 million variants obtained from this particular case. And then when filtering for only good quality variants, this number is reduced to 4.6 million. And then when reducing or removing all the variants that are present at a high frequency in the healthy individuals, this is, as you can see, the major step when reducing to reduce the number to around 4.5 hundreds of variants are expected to be functional. And then if we focus on the ones that are only expected to be homozygous, because in this particular case, the expectation due to the consanguinity with the family, we suspected that the variants are homozygous, and this reduced the number to 21. And then based on the association with the symptoms, we focus on genes that are known to harbor those symptoms. Then we ended up with only 3 candidates, and based on literature and further analysis and the severity or the scoring of the mutation, we ended up with only 1 variant that is eventually present in this gene, ARSA, that is known to associate with metachromatic leukodystrophy, perfectly explaining the symptoms. And this is another example of a dominant disorder. And as you remember from one of the previous slides at the beginning, a dominant disorder meaning that there is only a single nucleotide or a single mutation that could cause the disease. In this particular case, we have again an individual with microcephaly, seizures, motor delay, and leukodystrophy. And starting with whole genome sequencing, we had about 4.5 million variants. And this is then using more or less the same steps as mentioned in the previous slide. The only difference is that at this step, we excluded the homozygous variants instead of including those. So we focused only on heterozygous variants. And on top of that, since a dominant disorder was suspected, we also focused on genes that are only known to cause the disease in an autosomal dominant manner. And then when using the HPO match, we ended up with only 10 candidates. And by further scrutinizing this variance in those identified at this stage, by looking at how they are expected to be pathogenic, we ended up with 1 top candidate gene, which is known to cause lissencephaly 9 with brain stem malformation, which is also explaining the symptoms very clearly. And finally, I would like to finish with this slide. And this is an example from a paper that was published back in 2015. This is an example where we had done whole genome sequencing of a quadro, meaning we had the parents, the affected patient, and an unaffected sibling that all were sequenced by whole genome sequencing. By using more or less the same filtering strategy, we started off with a union total number of variants over 6 million. This is a combination of all the different variants identified in all the individuals from the family. And by the filtering, we use 2 strategies. In this particular case, the disease is known to be an autosomal recessive disorder, autosomal as well as X-links, by the way, I have to mention. So we first use the recessive mode of inheritance to identify genes that are expected, that follow the proper segregation. By using this proper segregation, we finally only ended up with 1 candidate because we could use the unaffected individual to remove all the variants that are not following this segregation. However, this candidate turned out to be not the disease-causing variants. And because of this, we changed the mode of the filtering strategy to also look into different mode of inheritance. which is a dominant mode of inheritance. And by using this approach, we ended up with 2 candidates, of which one we could validate as well as functionally test. And therefore, we were able to identify a novel disease-causing mutation in a novel gene with this disease that hasn't been described before. And this example shows us that actually, when you consider the mode of inheritance during the filtering, and you are not able to identify the disease-causing variant, you should always broaden your spectrum and also consider a different mode of inheritance that could cause the disease. And with this, I would like to conclude that it is important to know that even a single substitution can cause a disease-causing variant of a severe disease. It's not necessarily the larger the mutation, the more severe the disease. And accurate and comprehensive clinical description is critical for a correct diagnosis. It can happen that you may have different diseases present in a single individual. And therefore, the more and the accurate the description of the phenotype, the better it is to diagnose a patient. And also always consider different modes of inheritance in case the disease-causing variant is not identified in the first place. And finally, I would like to add that the inclusion of additional family members is also very critical to identify disease-causing variants, especially when you are dealing with a gene that hasn't been yet described to associate with syndromes or symptoms at the current stage based on the available literature. And the more individuals you have in a single analysis, the better it is to identify those novel genes. And with this, I would like to thank you for your attention, and I would be happy to take questions. Thank you.
Thank you very much, dear Najim. As always, very insightful, and thanks a lot for the presentation. Thanks a lot for preparation. Now, I see we already have Dr. Volas-Krajina here to help us out with the questions. There are already some questions coming from the audience. I'd leave it up to you guys to answer questions and discuss it.
Thank you very much, Nadim, for your great presentation. There are a lot of questions and comments from various countries. So for sure, we will not manage to answer all of them, but we start. So we have a question from US. It seems easier to detect substitutions rather than deletions or duplications. Could you please explain why?
Yes, so the reason why it's easy to identify substitutions, as you remember from one of the first slides, the first step during the analysis of the raw data is trying to align the data that is obtained from the sequencing to the reference genome. And as you could see, it's much easier to see a difference when there is only a single nucleotide difference compared with the reference. However, when it comes to gross alterations like duplications and deletions, I'm not even talking about the other complex rearrangements. What is usually required is not just looking at differences at that nucleotide level, but it involves a different type of analysis, which actually it needs to do a quantitative analysis, meaning the whole genome needs to be binned in certain regions, and then you need multiple samples to actually compare a sample of interest to an average of multiple samples to try to identify differences that could be explained by just comparing your sample to an average. So it involves a different kind of analysis, which makes it a little bit complicated. However, there are different tools that can do this analysis, and it is extremely developing in a way that it's much easier to identify currently than previously. And this is why whole genome sequencing, if I may add, is one of the proper approach to identify this type of alterations, which are usually difficult to identify with enrichment-based procedures like exome sequencing or panel sequencing, because they involve a biased approach of doing the sequencing.
Thank you very much. We have a question from Saudi Arabia. Why is it different sending the same sample and doing the same technology to different laboratories, and sometimes you get different results?
This can be explained by So during the complete procedure, there are different moving parts. One of the first is how the DNA is isolated. The way the DNA is isolated can have some minor changes. However, I'm not expecting this part to be the major contributor. And the next part is how the sequencing is taking place. So there are different sequencing platforms, and each sequencing platform can have its own biases. However, the majority of the labs that do sequencing nowadays is based on a single sequencer, so I don't expect that part to be contributing as well. But before the sequencing is obtained, there is one part where you actually go from DNA to generating libraries. And there are different kits nowadays that are used to generate these libraries. And one of those steps during the generation of the libraries, they involve the amplification of the DNA, and depending what kind of enzymes are involved, they can introduce some artifacts. And I think that lastly, where some difference can take place is at the level of the DNA analysis, because although I showed a workflow on how you go from DNA to the final report, when it comes to the analysis, there are different types of tools and metrics that can be used for the procedure that does the alignment to the reference genome and subsequent variant detection. So there are too many different components into the whole workflow that could explain one of the many differences that can be present.
Thank you very much. There is a question from Spain. If you receive the sample without any clinical characterization, can you still issue a medical report?
That's a very tricky question because, first of all, what is very critical when it comes to a medical report is the fact that you need to have consent to issue a report, which is a medical report, because you need to know whether the patient wants to know whether there is something in its genome that could be causing any symptoms, yes or no. So if that information is not available, it is very difficult to issue a report. And I'm referring to specifically incidental findings. If a person, for instance, sending a sample and may have a high risk of developing cancer, for instance, would this individual be willing to know this information? And this is very difficult to tackle. However, when it comes to having the possibility to identify the variants, the answer is yes. But whether we have the ethical obligation to present this data, that's more an issue that it's on an ethical level. We can detect the variants, but we don't know whether it needs to be reported. And this is entirely to the patient that should be well informed by the physician who eventually took the sample for sequencing.
Thank you. Is there any need to do PCR before doing NGS? Could you please comment on that? There's a question from India.
So there is no need if there's sufficient DNA. If there is sufficient DNA at the start, I would always urge to avoid doing any PCR. Although the majority of the labs nowadays do include a PCR step, especially when the DNA content or the DNA yield is limited, just to make sure that you would get the library that is analyzable.
Thank you very much. Could you please comment on nanopore sequencing? Do you use it or do you plan to use it?
So I use it personally, nanopore sequencing. It's an excellent technology to actually identify specifically structural variants because it has the advantage that it can sequence fragments which are megabases in length. The technology that we are currently using at Arsensus is the most widely used technology, which is actually based on shorter read sequencing. And by short, I mean 150 base pair read length, which is per end. So in total, we obtain about 300 base pairs of sequence data. So this technique is proven to be well in identifying single nucleotide variants as well as structural variants. However, there are some complex structural variants, especially in repetitive sequences, which are very difficult to identify and detect with this short read sequencing. Nanopore sequencing currently it has its advantages. However, the disadvantage of nanopore sequencing is the identification of subtle variants, because it still has a high error rate. So it can be used to identify structural variants, but it's not as powerful in identifying single or subtle variants that are usually known to be disease-causing.
Thank you. Your turnaround time in that census is amazing. It's 20 working days. How long does it take you to do bioinformatics?
Bioinformatics, once the data is available, it's so bioinformatics consists of multiple stages. The first stage is actually going from the raw reads, as I have shown, all the way down to the variants that are present, which are different than the reference. So that stage until actually the annotation, so getting some context to the variants, that can take up to 6 to 8 hours, depending on the machines that are available that are working in the background with this analysis. And then the final step is actually the interaction with the data, with these annotated variants by the clinicians, the clinical geneticists. And this step can vary a little bit. It can, in principle, take maybe up to 1 day. But in principle, when you have a very clear case, the symptoms are clear, the analysis can take place in 1 hour. From the analysis, from the data availability from the sequencer, all the way to the actual report can be up to 7 hours.
Just a note, the previous question was from Pakistan. Thank you very much. So we have a lot of questions. Is your laboratory accredited to do diagnostic testing?
Yes.
And what type of accreditation?
So maybe this is something that you can answer.
Yeah, we have ISO accreditation to do laboratory testing. Thank you very much. Why DNA testing is so expensive?
Well, I would assume that the question would be why DNA testing is so cheap, because I think it's the other way around. If you consider all the different steps that are involved, I think it's currently relatively cheap, because eventually one who takes the test needs to consider the entire workflow and the extent of knowledge that you obtain from DNA sequencing. And I'm specifically referring to whole genome sequencing, because this is data that is obtained that eventually can, on the long run, once the knowledge is there, we can use the information from this one test of whole genome sequencing to also predict specific symptoms that can be actually alleviated or prevented just by having this knowledge beforehand. So if you ask me, I would say it's actually a cheap test.
Thank you. And I've taken the last question. How do you know if the gene is causative if it wasn't reported before?
So that's a very good question. And this is the reason why I ended up with the last slide. And in principle, once you have stumbled upon this gene in the first place, there are different levels of evidence that is required to eventually say whether this gene is indeed causative. One of which is whether, for instance, this gene is coding for a protein that is closely related or closely interacting with yet another protein that is already associated with a disorder. So this could be a part of the evidence. The other evidence can come from, for instance, animal studies, where this gene has been abrogated in an animal model, and then this animal model is analyzed, and the symptoms are investigated, and they can compare whether the symptoms that are present in this animal are similar to those identified or manifesting in the individual that has been sequenced. And finally, one of the most important part is whether, for instance, this variant is identified in a family where multiple individuals has been affected, and whether this variant is indeed segregating with these individuals that share the same symptoms. So there are different levels of evidence that needs to be looked at before one can actually identify whether this gene is causative. And one of the most critical parts, which is usually depending on the lab that can do experiments, this gene can be actually made defective in a lab, in a dish, and then these cells with this defective gene can then be assessed to see whether indeed by abrogating the function of this protein, it indeed would result in the symptoms that are identified in the individual.
Thank you very much. So with that, I would close the question session. For sure, we didn't answer all of them, but we are happy that you contact us via email and we will answer your questions.
Perfect. Thank both of you very much. Super exciting listening to you. Cool.
Thank you.
Now, let me wrap up the session. Just two more small things. First of all, if you enjoyed it today, of course, you're super invited to join our next webinars on the 13th of April. Dr. Gabriela Opra, she's Chief Data Officer here at Ascensus. She will talk about improving my health based on my genome. So it's also a little bit more for healthy people, so to say, how we can use it in general to improve our health. And then we have the second part of our new webinar series, Unexpected Diagnosis based on whole genome sequencing, where Professor Dr. Arndt Rolfs himself will show you a new case from a real life scenario and see how we managed to solve it. Now, the last 2 points. For anyone who is interested in the CME certificate, we will post into the chat the link to this web formula, which you will find. We would like you to fill it out. And please be careful to fill out all the information completely, also with the full name, etc., because this is required in order to appoint the CME points for you. So you will find that link down below in the chat. Once you fill it out, we will receive the information and organize the certificate for you. And that's basically it. Now, from my side, the last point I would like to say is thank you very much. As always, it's a pleasure seeing how big the interest is, having more than 200 people from all over the world joining us. We will keep it up, make sure to continue evolving our webinars. If you have any feedback, any input, feel free to message us at any time. And it was an honor having you, and thank you guys. Take care.